I know you went through a lot of steps with a lot of sequences, and the whole time you're probably thinking, why am I doing this? So let's finally come to that question. So our goal with the PCR-based project is to test for contamination of your bacterial culture that you've been passaging for all this time. If you did contaminate it, the most likely source of contamination is your own hands, right? Your own skin microbiome. And that's how we chose the species that we're using as examples when we're testing for contamination. The Acinetobacter baumannii and the Staphylococcus epidermidis. So we know a lot about the skin microbiome from genomics research. These are some really cool graphs showing what the average skin microbiome looks like at all different sites around your body. The different colors represent different phyla of bacteria. So very broad categories in terms of bacteria that are quite different from each other in their features. And so I hope you can appreciate just by looking at it that the colors vary differently in different parts of your body, which is really cool, right? Like the bacteria that live on your skin, on your face, and in your nose are quite different from the bacteria that live on your skin, on your hands, which are different from the ones between your toes, right? So kind of amazing to think about. But they're looking at these colors, right? We're thinking about hands, right? So if we look at palms, for example, and really everywhere on the arm over here, we have a lot of this green color. And this is proteobacteria. And so our Acinetobacter baumannii is a member of this phyla of bacteria. And it's kind of our representative of this phyla. So these are gram-negative bacteria, meaning they have just one membrane on the outside of the cell. And so that's kind of our representative for this category. Another really, really common skin organism is Staphylococcus epidermidis. It's literally named epidermidis because it lives on your epidermis, your skin. Staph is a member of the Firmicutes phyla, and it's this red and orange. And so you can see those wedges here on the skin and lots of them on your feet. Over here, and lots of them on the back of your neck for whatever reason. But they're kind of everywhere. If you take micro, we'll isolate bacteria from your skin and identify them, and we see lots of Staphylococcus species, as you would expect. And so they're going to be kind of our representative of the Firmicutes, but also, as you'll see, kind of our representative of everything that's gram-positive. So those bacteria with two membranes. The vast majority of the bacteria that live on your skin are gram-positive, so it's kind of a representative of a lot of species. So that's where those two came from. So the next question is probably, why am I looking at a 16S rRNA sequence? Like, why this part of the genome? We talked about it very, very briefly in class when we were talking about the kinds of research we do for genomics. And it's because this is the sequence that we use to identify species. And again, our goal with this PCR project is to identify what species are in your sample, right? We're hoping it's straight up E. coli and nothing else. But we're going to use the 16S ribosomal RNA sequence to ask that question. So sequencing 16S rRNA is how we came up with those pie graphs we were just looking at of what kind of bacteria live all different parts of your body. We identified them by sequencing this gene. How do we do that? Well, the gene has sections that are conserved in all organisms, all bacteria. So high sequence identity, which are these peaks on this graph and represented in white down here. And then it also has regions where the sequences are really unique. So where the sequence identity drops to very, very, very low, and we call these variable regions, and they're represented in blue down here. And so essentially this gene has regions that have parts of the sequence that are unique to every different bacteria and parts of the sequence that are same in all the bacteria. And that's really helpful for us, right? Because you can put your primers when you're trying to sequence everything in these conserved regions where it's going to hopefully bind to the genome of every single different random bacteria in your sample. and copy, right? And then you can sequence in between. We're taking a slightly different strategy because we're doing only PCR. And so hopefully what you noticed as you were predicting your PCR products is that some of the primers anneal to all the sequences and are going to give you a product for everything. And we'll use those kind of as our test for, is PCR working? Is there bacteria in our sample? It should be a yes, kind of our positive control for the process. And then other primers are more distinct, right? They're binding to these variable regions and therefore only binding to some species genomic DNA. And so we'll be able to use those primers to test for, are these other species different from E. coli in here, right? Are these sequences that are clearly not E. coli sequences in my sample? So to look at that a little bit closer, here is what we call a sequence alignment. So these are 16S rRNA sequences from all these different bacteria. So I have a lot more on here. I did this when I was trying to choose some options. So here is our DH5 alpha E. coli EC. Here's our Abomanii, lovingly called A-bomb, and our Staphylococcus epidermidis, which I have abbreviated as epi. So those are the three that I chose out of this big long list as kind of classic skin bacteria, but I wanted to look at rRNAs from lots of different species. So their sequences are aligned, which means that this computer algorithm took all those sequences that I told it and matched them up as best it could. And everywhere that there's a star on the bottom, all the sequences are the same. And so these stars are our conserved regions, right? So our white areas on this graph where the sequences match in all the different kinds of bacteria. So this covers a fairly wide spectrum of different kinds of bacteria. And despite that, right, like if you look right here, they all have the exact same sequence in their 16S right here. If you scroll down just to the next row, right, there's no stars here. And you can see there's a lot of diversity in the sequences right here, right? This would be one of our variable regions, right? Where the sequence similarity is low when you compare across lots of different bacteria. And so you'll see that kind of back and forth pattern as you scroll through, right? Conserved area, less conserved area. Okay, if we keep going down, very not conserved area here. Keep going down. This row, quite conserved, right? Lots of stars, sequences looking very similar. This one, almost nearly identical in this whole little region, right? So this is clearly a conserved area. And you'll see that pattern as you go through. So what I highlighted on here is our primers. So right here at the very front, our first primer, which I believe is number 23, our forward primer, you can see is in a region with lots of stars. So this is a very conserved region. We wanted this primer to stick to every different kind of bacteria's genome sequence. And we see that it should do that, right? Because it's in a region that's conserved. There's a couple of little differences, which we saw when we were attaching it to the Staph epi sequence, right? It's got one little difference here. But for the most part, they match. And so this primer should be able to bind to all bacterial 16S or RNA sequences in the genomic DNA. Continuing along, here's our next primer. I believe this is 24. And it's highlighted only in the E. coli sequence. And that's because there's diversity in the sequences here, right? There are a couple other sequences that do look very similar. The two right above it are identical, right? This is identical, and this is identical. But these are actually other E. coli. So I was just curious how other E. coli looked. So we're using DH5 alpha. These are just two other E. coli strains. And so this sequence is conserved in all E. coli. It's also conserved in this species here. This is Vibrio cholerae, another gram-negative bacteria. So not so far away on the kind of similarity tree of bacteria. If you look down here, here's our Acinetobacter baumannii. And it's got some similarities, right? This whole half of the primer all the way to here, right, is identical. But this half, not so much, right? And so in your prediction, you probably said, oh, primer 25 is going to only bind E. coli, and it's going to be specific for E. coli. Is that actually going to happen in real life? I don't know, because this sequence in Acinetobacter baumannii is still pretty similar because it's not so far away on the bacteria family tree, right? But all of our gram-positive bacteria up here, kind of on the top half, wildly different, right? So at the minimum, this primer will probably be able to at least distinguish between gram-negatives and gram-positives. We keep scrolling down. Here's another primer, right? I think this would be next on our list, 25. I think I numbered them in order as I went down the gene. Don't quote me on the numbers. This one is in a region with stars, right? Identical in every single bacterial species in this alignment. So this is a primer that's not going to be specific for any one species. It should work in everywhere and kind of function as our positive control for, can I do a PCR correctly and it work? Because this one should work no matter what is in your sample. Hopefully you predicted that in your chart. Continuing along, here's a primer that I have highlighted in the Acinetobacter baumannii sequence. So that's the only one where we've got an exact match. Looking at all the species in this alignment, we also have a match almost perfectly here. It's got a couple of differences there towards the end. This is Pseudomonas aeruginosa. So Pseudomonas is the most closely related to Acinetobacter, generally speaking. Also kind of could be a part of the microbiome, some Pseudomonas species. So it would be okay if we can't distinguish between these two with this primer sequence, but look how different E. coli is. So our primer really shouldn't stick there. And look how wildly different all our gram-positives are up here on the top half. So hopefully what we want is for this primer to tell us, right, to only bind other gram-negative species, right? Not our E. coli, like contaminating gram-negatives. And so we're predicting that's how it's going to work. Again, TBD on if it actually works. But our goal with that one is to find contaminating gram-negatives. And then continuing on down to our last one, which is way down here. Here we are. So I have this sequence highlighted in the staph epidermidis sequence because that's where it matches perfectly. But you can see that in this top half of our alignment, all these gram-positives have really similar sequences, right? So really this primer is probably going to amplify anything gram-positive, which is kind of great, right? Because we don't want anything gram-positive in our sample. We want only E. coli, which is gram-negative. And so it could be a great primer for just testing, do I have contamination with a gram-positive? And again, the vast majority of the bacteria on your hands and in the environment are gram-positive. And so this one can be really helpful for our contamination check. And again, wildly different from all the sequences on our bottom half, which are gram-negative guys down here. So hopefully that one will be a great primer to test for contamination. So hopefully that explains some of the logic behind this process and what I was doing as I picked these primers. Again, TBD, we'll see if they work the way we're hoping. But answer those final few questions, make predictions about what products you're going to see if your sample is contaminated or if it's not contaminated, so that you fully understand what you're looking for when we go and do these PCRs to look for contamination.